Hello everyone, I am back with my new video with the topic animal tissues. Okay, before starting today's topics, let us quickly recapitulate everything. Okay, so the first thing comes is who was the first person to term the word tissue? It was Bichat. Father of histology is Bichat, and there was another scientist, Marcello Malpighi, who was father of microscopic anatomy. Then comes what are tissues? Basically, it is a group of similar cells performing a similar function with the intercellular spaces. Then next comes types of tissues on based on their functions. Basically, there were 4 types. Okay, first comes is epithelial tissue, then comes muscular tissue, then comes nervous tissue, and the last one is connective tissue. Okay, so first we are going to study about epithelial tissue. Okay, let us know about them. Epithelial tissues are basically avascular in nature. Avascular basically is that they do not have blood vessels, lymph vessels, etc. Okay, so how do they get nutrition? They get nutrition with the help of basement membrane. Now what is basement membrane? It is formed by the help of 2 types of tissues. Okay, epithelial tissue and connective tissue. Epithelial tissues forms the basal lamina made up of glycoprotein, and connective tissue forms the fibrous lamina, which is made up of what? Protein fiber and hyaluronic acid. Okay, now this basement membrane is highly permeable and non-cellular. It is non-living because it has no cells. Sorry. Now next comes modifications. There are 3 types of modifications we are going to study, that is microvilli, cilia and stereocilia. Microvilli are basically known as brush bordered epithelium. It is found in the parts of our body like in gallbladder, PCT of nephron, and also in walls of small intestine. It helps to increase the area of absorption also. Okay, and it is non-motile and non-contractile. Next comes is cilia, or also known as cynocelia or ciliated epithelium. It is found in the areas where we have to provide direction to something. Okay, like in fallopian tube we have to provide direction to the egg, and also in uterus, which provides direction to the sperm. Okay, these are some examples. Now in trachea and bronchiole there are also ciliated epithelium, and also in ependymal epithelium. I have also discussed about these things. You can check my previous video. Next comes is stereocilia, stereociliated epithelium also called as. Okay, it is found in the areas vas deferens, epididymis and macula and cristae of internal ear. Now types of epithelium are basically, before that we are going to discuss about the junctions. There are 4 types of junctions: tight junction, gap junction, interdigitations and desmosomes. We have discussed the properties or functions of it basically. Now let us start with today's topic, which were types of epithelium. Now there are 2 types of basically simple epithelium and compound epithelium. First comes simple epithelium. Based on the shape, it can be of 4 types: simple squamous epithelium, simple cuboidal epithelium, simple columnar epithelium and pseudo stratified epithelium. Okay, now first is simple squamous epithelium. It can be also of 2 types: pavement epithelium or tessellated epithelium. Its function is basically filtration and diffusion. Wherever you will see filtration and diffusion, you will find simple squamous epithelium tissues. Now let us know about their examples. First one is alveoli or air sacs of lungs. Second one is Bowman's capsule. Third one is thin part of loop of Henle. Fourth one is outer surface of intestines. Fifth one is endothelium of blood vessels. Sixth is endocardium of heart, and seventh is mesothelium or peritoneum. Okay, now let us see the diagrams which will help us to visualize even more. See, in the alveoli or air sacs of lungs, this is a basically structure where pneumocytes 1 is present. Pneumocytes 1 are present in 95% of the area. Okay, this is basically structure of alveoli. Now next is Bowman's capsule. See here, Bowman's capsule. The cells present there are known as podocytes. you must remember these names, pneumocytes 1, because we are going to study pneumocytes 2 also, that is why I am telling. Podocytes in Bowman's capsule. Now thin part of loop of Henle, see this is thick part of loop of Henle and this is thin part, okay. So simple squamous epithelium is present in thin part of loop of Henle, okay. Now as we discussed about outer surface of intestine, endothelium of blood vessels. In endothelium of blood vessels, tessellated mesodermal epithelium is present. See this blood vessel and tessellated type of epithelium is present. Next is endocardium of heart. Here also tessellated mesodermal epithelium is present, okay. Now next comes is mesothelium or peritoneum. See this is our body basically, this is the mouth and this is the anus. The pathway from mouth to anus is also known as gut or alimentary canal. Now we are going to take a cut and to look through its dissection. So we will get the structure as this. So here and here, both of these areas, it is simple squamous epithelium, okay. This inner layer is formed with the visceral epithelium and this outer one is formed by the parietal epithelium. Both together are known as mesothelium, okay. This is also, this space, empty space is filled with coelomic fluid and also called as body cavity, okay. Now next comes is simple cuboidal epithelium. Its function is absorption and secretion, also known as germinal epithelium, because it gives birth. Let us see the examples. First one is alveoli, air sacs of lungs, pneumocytes 2. There we have discussed pneumocytes 1 and here it is pneumocytes 2. So simple squamous epithelium is present in 95% and simple cuboidal epithelium is present in 5% or pneumocytes 2, okay. Second one is follicles of thyroid gland. See in thyroid gland we are taking a single cell, this is called as follicle, okay. So here cuboidal epithelium is present, this thyroid follicle, okay. Next is acini of pancreas. In the pancreas we are taking a small part of it, okay. So we will get a structure like this and this is the simple cuboidal epithelium, it is filled in the whole space, okay. Next comes is secretory unit of sweat gland. Now this is our sweat gland and basically this is the secretory unit and this is the duct, okay. So its function is secretion also, so it secretes the sweat. Next is thick part of Henle's loop and DCT of nephron. DCT is basically distal convoluted tubule, okay. Now next are iris, choroid, ciliary body and retinal epithelium. All of these four things are present in eye and you can find simple cuboidal epithelium cells. See this is our structure of eye. Outer portion is known as sclera, then comes choroid, okay. Then inner one is retinal epithelium and this is iris, the elongated one, and this is the ciliary body. We will discuss about all these functions in the coming videos, okay. Now let us see the next thing. Now there are modifications in cuboidal epithelium also. So two modifications are basically there. Simple brush bordered cuboidal epithelium. See here also brush bordered kind of thing is there. It increases the rate of absorption and also surface area, okay. It is found in the PCT of nephron because from there we have to absorb a lot of things. Second one is simple ciliated cuboidal epithelium, okay. See this is cuboidal epithelium and this is cilia. So it is found in collecting duct because cilia helps in providing direction to something, okay, whether it be upside, downside, anywhere, okay. So that's all for today. Thank you.